Hello, my name is Britannia Campbell, and I will be discussing the dynamics of Th1 or Th17 responses and antimicrobial pathways in leprosy skin lesions. And this is my research paper. Now let's get into the actual research paper. So in this presentation, we'll be going over how the immune system reacts during a reversal reaction in Leprier. We'll also discuss how this type of shift affect the body fighting microbiome in Leprier and how does this affect future research going forward. So that's just how this paper breaks down each step, which I will be going through with you. Leprosy exists on a clinical spectrum. On one end, we have tuberculoid leprosy, and on the other hand, we have lepromatous leprosy. Now, tuberculoid leprosy is where the patient exhibits strong cell-mediated immunity and can contain the infection very, very well. But on the other hand, with lepromatous leprosy, the immunity is weak and the bacterial load is extremely heavy, or in other words, is just too high. Now, reversal reactions can occur when a patient suddenly begins to mount a much stronger immune response. Now, this can be beneficial for bacterial clearance, but it can also cause inflammation and even lead to nerve damage. Now, understanding the mechanisms behind reversal reactions is important both clinically and scientifically, and you will see this as we go along in this presentation. Now, in this paper, the author's main goal was for us to understand what exactly drives the immune system shift during a reversal reaction. Now, they focused on three main topics, which was identifying the immune pathways that become activated during reversal reactions. Secondly, it is categorizing the antimicrobial genes that contribute to bacterial killing. And then lastly, it was discovering new antimicrobial proteins that humans naturally produce during reversal reactions. Overall, they just wanted us to map out how immunity becomes effective again, even with leprosy. And to put this in their own words, in this study, they sought to uncover the dynamics of innate and adaptive antimicrobial mechanisms at the site of disease by investigating the dynamic changes in the reversal reaction transcriptosome in paired skin biopsy samples collected from patients before and at the onset of the reversal reactions. The first major result was the discovery of a large antimicrobial gene during the reversal reaction. Now, out of 400 genes that changed significantly, 200 were upregulated. And among these, the researchers identified a 64 gene antimicrobial signature. Now, the expression of these genes were inversely correlated with bacterial burden, but this shows that the reversal reaction is not just inflammation based, it is coordinated. It is a coordinated antibacterial response as well. And this is to put it into simple terms. Higher expression equal fewer bacterial presence. The second key finding here involves how antimicrobial programs are regulated. Now, the study identified four major cytokine drivers, or as you can see here on screen, four major upstream regulator. All four of them are listed here, which are two innate and one Th1 and one Th17. Now, all of these cytokines collectively regulate most of the antibacterial gene signature. Interestingly, the single cell analysis showed that Th17 cells were producing and Th1 response, in this case INFY, which is typically considered a Th17 phenotype, playing a major role in reversal reactions. Now, another notable finding in this case is that antimicrobial genes weren't expressed only by immune cells, but they were also expressed by keratinocytes, myeloid cells, fibroblasts, endothelial cells, and T cells. All of these play at least a special part in expressing the antimicrobial genes as well. With machine learning, the authors were able to predict a few things. In this case, they were able to predict which reversal reaction upregulated genes might encode antimicrobial peptides. In this case here, they identified 41 candidates with structural features suggesting the ability to disrupt bacterial membranes. Now, from these four, four of them were selected, as you could see on the bottom of the screen. We have S100A7, S100A8, CCL17, and CCL19. Now, not all of these had previously been recognized as antimicrobial, which make them extremely, extremely interesting and important. Now, in this test, which I find to be the most interesting, is the functional testing. It produced some of the most compelling results. All four of the proteins that we discussed on the previous slides, which were S100A7, S100A8, CCL17, and CCL19, all four of them showed significantly reduced leprosy visible inside the human macrophages. And additionally, in bacterial cultures outside of the cells, the proteins still exhibited direct antimicrobial activity. That is exceptional. And that's really the mark that I think the study was trying to hit. Now, the electron microscopy also revealed the mechanisms that the bacteria showed, and these included membrane rupture. It also showed disorganization and structural collapse. This just goes to confirm that these proteins act as a genuine antimicrobial effector as well. The antimicrobial signature observed in the reversal reaction patients was also seen in typical tuberculosis patients, and these patients naturally had stronger immunity. Now, on the other hand, patients with lepromatous leprosy had very or usually show very low expression of these genes. This type of correlation validates the signature and shows that the study accurately captures the immune differences across the disease spectrum itself. So the main outcome of the study is that reversal reactions represent a coordinated multicellular activation of the Th1 and Th17 immune system pathways that helps to restore the antimicrobial capacity. Now, this isn't just any or information or regular information, but it is a deliberate and effective antibacterial program that involves cytokines, epithelial cells, myeloid cells, and direct antibacterial proteins, all effective in the response to induce multicellular antibacterial programs. This study has major implications for future research. It identifies previously unknown microbial proteins that could potentially become therapeutics. Now, second, it highlights the importance of the Th17 cell in controlling intercellular pathogens like leprosy, as we have been seeing here. Lastly, it opened doors for new therapies and strategies to enhance the patient's own immune system rather than relying on outside sources or any type of medication when the patient's own immune system could be doing the work for them. And I think anyone would much rather that be the case. In summary, the study used a powerful paired sample approach to understand the immune dynamics of reversal reactions. It identified a 64 gene antimicrobial program that correlates with the bacterial reduction, discovered new human antimicrobial proteins, highlighted a central role for Th1 and Th17 immunity, and overall, the work provided a valuable insight into how the body regains control over leprosy and offers multiple promising avenues for therapy development in the future. And maybe we could overcome the use of antibiotics for these things, but take on a more natural course of how to solve these issues. This is the end of my review. Thank you all for watching, and I hope you enjoy the rest of your day.